Hello, everyone, and welcome to today's webinar, Accelerate Discovery and Development by Optimizing the Protein Engineering Workflow. This webinar is part of the 7th annual event in the Drug Discovery and Development Virtual Event Series. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You will also notice that the speaker has provided a survey. This survey will be available during both the live session and the on-demand period. In addition to asking questions, we encourage you to participate in 3 poll questions throughout the speaker presentation. Simply click on your answer when the poll questions are displayed. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Claudia Chiocchini, Manager, Research and Development at Thermo Fisher Scientific. For a complete biography of our speaker, please visit the Presenter tab from the menu on the left of your screen. Claudia, you may now begin your presentation.
Thank you for giving me the chance to be here. My name is Claudia Chiocchini. I'm an R&D manager at Thermo Fisher Scientific, based in Regensburg, GeneArt in Germany. And today I'm going to talk to you about strategies for boosting your antibody production in mammalian cells, and also illustrate some of our tools to facilitate, accelerate your research. At Thermo Fisher, we are proud of our mission, which is to help our customers to make the world healthier, cleaner, and safer, which was particularly true in the past 2 years of pandemic, where at GeneArt Thermo Fisher, we are able to synthesize hundreds of genes that served as a template for the synthesis of anti-COVID vaccines. So today I'm going to quickly go through, give you an overview about the GeneArt synthesis services, and then talk a little bit about the key application of biosynthetic genes. And then I will explain more about our gene optimization and how it works, the proprietary algorithm that we have at GeneArt. And then I will talk about our protein expression services that can facilitate and accelerate your research. And at the end, I will explain with some customer examples how we can achieve the higher protein yield. Okay, now looking at an hypothetical workflow, everything starts with a simple amino acid sequence or DNA sequence that you can provide to us. And this sequence is going to be optimized for maximizing expression in a certain host organism. And this organism is normally mammalian cell when we deal with antibodies, of course. So the gene optimization is the one that I will explain in a little bit more detail. It's the first step. And then we go with the physical gene synthesis. Of course, we can also do gene variants or entire gene libraries. And this can be cloned into a vector of interest. It can be a vector that you choose from our in vitro gen selection, or just the vector that you use in your laboratory. And plasmid production can occur, of course, at many different scales. The next step, natural step if you go with antibodies, of course, is the transfection of our cell lines, and then antibody production and purification.
We would appreciate your participation by answering a few polling questions. To record your answers, click on the option on the screen and select all that apply. Our first polling question is, which aspects of your antibody engineering workflow would you consider outsourcing? A, gene synthesis, B, expression, C, purification, or D, characterization. I will give you a few moments to select your answers.
Different application of synthetic biology is, these are of course manifold. So we talked already about vaccine development, but this is not all. So what you can do with synthetic genes is of course also to develop therapeutic antibody and antibody engineering, or go for drug discovery, or enable new biotechnologies. Now looking at gene optimization, why is gene optimization so important? It is intuitive that the codon usage plays a crucial role in protein expression. And this is particularly true for antibody expression as well. So looking at the codon usage, the codon is of course degenerated, the genetic code is degenerated, so you can use different DNA sequences to code exactly the same antibody sequence. And why would it do that? If you have an autologous gene expression, so if you have a mammalian gene expressed in a mammalian cell, so everything should be optimized by nature. But this is not 100% the case, because genes are not optimized for maximal expression. They are optimized for maximal regulation. So if you want to boost your protein expression, you need to do some changes at the DNA sequence level. And what we do with the gene optimizer is that this algorithm takes into consideration 20 different parameters. So it's not only about the codon adaptation index itself, but it's also taking into consideration GC content, the presence of cryptic splice size, secondary structures, or direct repeats, and all elements that indirectly influence protein expression. So in this slide, you see an example how the gene optimizer actually works. So what you see on the left side of the slide is a wild-type gene sequence from 5' to 3'. And this is usually composed by codons that are optimal for expression, or say favorable codons, which are highlighted in green, and less favorable codons, which are highlighted in red. So optimization is not a simple back translation. So it's not the replacement of the red codon by the green codon, but it's something more complex. So it's finding a trade-off between the codon adaptation index and the absence of the motifs I was mentioning before that are hampering protein expression. So on the right side of the slide, you see how the gene optimizer actually works. So all possible codon combinations are tested, but not over the entire sequence at once. So the algorithm takes a small window and checks in this window the best codon combination. The first best codon, it is locked down, and then the window slides from 5' to 3', and the operation of searching the best codon is repeated over and over. An extended window is also considered to check the codon combinations and also to look for the absence of the hampering motif I was mentioning before. So with this mechanism, many steps are done, and what you see in step number 6, this is our optimized sequence, which is of course containing optimal codons, but also codons that are less favorable. To prove the performance or the efficiency of our gene optimization, there was a study we did a couple of years ago, and this was based on 50 different sequences taken from the NCBI database, addressing many important proteins that are also drug targets, like membrane proteins, cytokines, transcription factors, or ribosome proteins. And these 50 proteins were taken and they were optimized. So we had a direct comparison between the wild-type sequence and the optimized.
Our next question is, would you like to speak with a technical sales specialist about options for gene synthesis and protein expression services? A, yes, or B, no. Please take a few moments to select your answers.
In this graphic is the comparison of different Western blot analysis, where you see that we consistently got a better result with the optimized sequence. Precisely, we found out that 86% of the sequences we were checking were giving a better expression compared to the wild type. In 10% of the cases, we had pretty comparable expression, And in the remaining percent, it was just 2 membrane proteins, we saw a slight lower expression compared to the wild type. This is probably due to the fact that the membrane protein were overexpressed in the cells and turned out to be toxic for the cells. So in this picture, you see what happens when we include antibodies in this observation. This is a comparison between wild-type M-optimized gene from 3 intracellular protein and 2 antibodies. And you see in the case of the antibody, is the increase of the expression is really clear. So we have a 12-fold increase of expression for the one antibody and 15-fold for the other. And the wild type is set as our reference. So now the next step was to check how the gene optimizer performs compared to other competitors, because of course there are a lot of competitors out there offering the gene optimization. And the good news for us was that we were checking now 5 different competitors, and our gene optimization was the only one which was delivering consistently better result of the optimized sequence compared to the wild type. So if you look at this at this histogram and take whatever protein, let's say number 3, you see that for some competitors this protein has a higher level compared to the wild type. But looking at the same competitor with another protein, they will get a negative result, taking the same gene optimization. So the message again, GeneArt Gene Optimizer software outperforms our competitor and delivers consistently good results with the optimized sequence. Now I want to talk to you briefly about our protein expression services. And I want to point out that those services are available to you, but you can replicate the same what we do in our lab, in your lab, but just using the same ingredients, let's say the same expression kit. So our protein expression services is based on expression in mammalian cells. We do have insect cell, SF9, and the baculovirus system, but many of our customers, the majority of our customers request expression in XP293 and XP-CHO cells. Now, looking at the power of this transient expression system, why is that so successful? And many customers go back to our kit in XP293 and XP-CHO cells. Well, the reason is the cocktail is perfect. So you have an expression media which is optimized for growing cell at very high density, and this is the Gibson media. You have cells that produce more protein per single cell and also adapted to grow at very high density. And then you have transfection reagents which are super efficient and are designed to deliver DNA to the cell in this high density culture. And this cocktail can even be improved and its potential is even higher when you combine it with the gene optimization that I was explaining before. So these incredibly boost the level of production of your antibodies. Now looking at the single components just quickly, so the Gibson production media is an animal-free media, and this is basically suitable for both the cell maintenance and for the transfection. So you don't need to switch medium when you go from the normal cell culture to the transfection. It is designed to grow cell at a very high density of about 18 million cells. And it supports very high expression yield. So if you look at the graphic and you see XP293 growing in different media and growing in this optimized Gibson media, you see that after 4 days, you work in a totally different range, that the cell density is very high, but also the viability stays really high. So now going to the cell lines, the cell line optimization is something that is done at Thermo Fisher. We do not develop stable cell lines at GeneArt, but it's something that our colleagues in US do. So the design of these cell lines, 293 and XP-CHO, is really done to cover all the needs of our customers. And they have, of course, slightly different features. So if you look at 293, for example, they can be passaged even 30 times, and XP-CHO only 25. They have different doubling time, with XP-CHO being a little faster than XP293. And they both produce protein, active, correctly folded protein at a high quality. And another difference is that the cell density is slightly different of those two systems. Now, when you look at the performance in producing antibody or different proteins, you see also that there are a slight difference between those two. So CHO is let's say, the best producer with an average of 3 grams per liter. And the XP293 is more robust, but it produces slightly lower titer with 1 gram per liter approximately. I have to say these data are, of course, just references, right? Because with your antibodies, you will have your own experience, and every antibody is different, and the level of expression is different, so you don't have to take these as a golden standard, but this is just a reference. And if you look at the common applications, well, 293 are preferred for biostructural studies, for example, or expression of intracellular proteins or membrane proteins, whether if you look at XPCHO, they are the best choice for expression of antibody at a high titer. So bispecific are normally done with XPCHO or even Fabs or regular full-length antibodies.
Our final question is, are you interested in getting a quote on a protein expression project for your future research? A, yes, or B, no. Thank you so much for participating in those polling questions.
Now, looking at our services, so we are very flexible. First of all, I have to say our service is fully customized. So you can come to us and we can express the protein you desire in the condition you want. So specific buffers, specific purification protocol. We take everything, but also we have services that are more streamlined. And one example is our small-scale, high-throughput production service. This is something we launched as a pilot in 2020, and we are constantly optimizing this offer. So at the moment, we are looking into automation of this process to make it even more reliable and traceable. So with this, we have two offers, the 96 well plate format and the 24 well plate format. In 96 well, we express a little less because it's just 1 milliliter, and with 24, we can go to 2.5, and this is the reason for the slightly different yield. So again, looking at average yield, we go with around about 300 micrograms for the very small scale, and 570 for the 24 well format. So those two pictures also highlighting two different features of the service. So on the left side, you see different antibodies and replicates of those antibodies. And on the right side, you see the same antibody producing replicates. And it should give you an idea of the consistency of the productivity. And if you look at the analysis, the standard is simple STS page chemosis staining, which we can do in reducing condition, where you would, of course, see the light chain and heavy chain, which you see in the middle of the picture, or in non-reducing condition, where you see the full-length antibody at about 150 kilodalton. HPLC analysis is not included in this service, but we put it there as a reference to show you that the antibodies are highly pure and the high percentage of monomeric state, which is important to many customers. Also, we did some analysis of the endotoxin level, and we see that this is acceptable mostly for many customers because it's below 0.1 per milligram of protein. Now, looking quickly at the general offer, as I said, we are very flexible in scale, so we start with the small scale, but we can go up to higher scale. So what is possible? So 35 milliliter, 250 milliliter is, I would say, the most asked scale, normally by our customers, and we are able to do around about 300 antibodies per week at this scale. And we can go up to 25 liter, which we would then do in a weight bioreactor. So looking at the purification methods, we are, of course, talking about antibodies. So it's the standard affinity chromatography. We normally use basically protein AG resins, but we can produce many different proteins, so also antigens. So any epitope tagged protein, you can take the normal his tag, strep tag, classical flag tag, or C tag. And we can offer downstream different services, not only the regular analytic services that you already know, but we can also eliminate the tag if you need it, adjust the protein concentration to a certain amount, even aliquot your protein, So whatever you need for your downstream application. And concerning the purification step, again, depending on the scale and depending on your needs, we can go with several steps. So affinity is the first one. We can do ion exchange, or we can have even a polishing step through gel filtration if you require this. Looking at the analytics, so the standard, as I said before, is STS page and Western blot, but we can also offer a capillary electrophoresis. This is something that is also very asked by our customers, that we use normally the lab chip, probably know from PerkinElmer. And then HPLC analysis, SEC HPLC to determine the aggregation state of your antibody or the percentage of monomeric state. We can go with normal mass spectrometry analysis, which we normally do to confirm the identity of your protein, and also endotoxin determination. So you can separately order one of these add-on to your service depending on what you need. And here you see an example of the HPLC profile at a SEC HPLC analysis where you have a monomer and also to some level of oligomerization. So now looking at the customer examples, I just brought 2 examples to show you how our service can really help you on in your lab to speed up your research and free your hands to do your actual research. One customer came to us with a special request to have a higher antibody titer. They were using XP293 and they were using it to produce proteins, so mainly antibody, for immunologic research, for cancer research. So our response was, first of all, to advise them to switch to the XPCHO system that normally provides higher titer. This was a first improvement, but they were still investing a lot of time in protein production purification. And so they decided to outsource the production completely to us, which made the real change because we could introduce our gene optimization that, of course, boosted their protein production even more. And second, they could save a lot of resources. So the estimate is about 2,000 FTA in a single year, which is a lot. And they had an antibody production which was 50% higher than what they had before. In this other example, the customers had a problem. They wanted to use the XP293 and not switch to another system, and they wanted to have a higher protein titer. So here the advice was to stay with our system. We also tested the freestyle 293 that didn't perform as well as the XP293, and then we switched from their vector to our pcDNA 3.3 vector, which even increases the expression. And then we combine this with the gene optimization. So the result is clear. You see the histogram is basically the pale blue bars where the expression is boosted. So in summary, if you decide to outsource your protein service to us, you will have a complete service chain. And what is really important to many of our customers, you won't have any transfer of electronic sequence information or even sample transfer from one site to the other. Everything can be done at our site under one roof. You will have a reliable performance due to the fact that we use best-in-class expression systems such as XP293 and SPCHO. And you can take the same system and replicate the results we get in the lab in your own lab. And you have a scalable and flexible service where we can really satisfy your needs in terms of whatever you have, different protocols, buffers, and different scales. And in the end, you will have also a transparent communication, what's happening with your project. You can contact our services and you can look into the progresses, and if there are any difficulties, any delays, you'll get to know it immediately. And we have active MSA with many pharma and biotech companies. So let me mention that Regensburg is also one of the Thermo Fisher sites committed to zero waste, along with other sites, of course, and 0% of our waste goes into landfill. So with this, I would like to thank you very much for your attention, remind you that you can reach out to us through our local sales representative. You can go to thermofisher.com, gene2protein. Thank you very much.
Thank you again, Claudia, for your time today and your important research. Before we go, I'd like to thank the audience for joining us, and I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until February 21st, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.